Welcome back, students. This will be the third video from chapter 2, and we'll continue to the end of this chapter. Module number 2 discusses the molecules of life. These are the main molecules found within living cells. This module covers a rather large list of keywords from the word bank as indicated here. When the question is raised as to what one finds inside living cells, the answers vary widely. Sure, everybody knows that living things are made of carbon because we're carbon life forms, but what does that exactly mean? In the simplest terms, it pertains to the composition of the chemistry within cells. The 5 learning outcomes for this module will help the student understand the previous statements. The first of these is to be able to list the 4 main classes of molecules found in living cells in the order of abundance. The second learning outcome requires students to be able to compare and contrast the chemical structural features of the 4 main classes of these biological molecules. The third learning outcome requires students to describe the role of condensation and hydrolysis reactions in the making and breaking of chemical bonds. The next learning outcome is to be able to differentiate between monomers and polymers. And finally, the last learning outcome requires a knowledge of the composition of cells. If one was to examine the contents of a typical cell, regardless of plant, animal, or prokaryotic, the general conclusion is indicated in this slide. About 70% of that cell would be composed of water. The balance, the remaining 30%, would be divided into the following molecules. The vast majority of the 30% would be proteins, 6% would be RNA, 2% would be sugar, 2% would be phospholipids, and 1% would be DNA. The remainder will be a concoction of ions such as sodium, potassium, calcium, and small molecules like vitamins and minerals. Please note the small error on this slide. Phospholipids are not classified as macromolecules. The word macromolecule is reserved for huge molecules such as DNA, RNA, proteins, and carbohydrates. Phospholipids do not attain these very large sizes. If water is excluded from one's calculations, it's evident that the vast majority of the remaining chemistry is based on carbon, thus the label carbon-based life forms. Also in chemistry, any molecule which contains carbon and hydrogen is known as an organic molecule. And as we'll see shortly, the 4 major types of chemical molecule found inside cells are all organic molecules. Table 2.2 presented in this figure just requires a little further explanation. This column here is telling you the percentage of the cell weight occupied by that chemical class, whereas the final column simply tells you how many varieties of that particular compound exist within that category. For instance, with water, 70% of the cell is water, and there's only one type of water molecule, H2O. This explains the value of 70 in this column and the value of 1 in the final column. Looking at the next row, in terms of inorganic ions, such as sodium, the total percentage they occupy in a bacterial cell is just 1%. But there are 20 different varieties, sodium being one, potassium being another, and calcium being a third, etc. Up to 20 different varieties. Continuing to read down in these rows, we find that the sugars occupy 1%, and there's 250 different types of sugars inside a typical bacterial cell. Under 0.5% of the total weight of a cell is amino acids, but there are 100 different varieties of amino acids and their precursors. Nucleic acids and their precursors occupy just under another 0.5%, and there are 100 different varieties of those too. 1% of a total cell's weight is fatty acids and their precursors, and there are 50 different types of fatty acids. And finally, at the bottom, we have macromolecules. Macromolecules are huge molecules made up of either amino acids or nucleotides or individual fatty acids and sugar precursors. And these macromolecules account for 25% of the mass of a cell, and there's over 3,000 different varieties. Living cells possess 4 large classes of molecules, namely polysaccharides, or polysugars, lipids, proteins, and nucleic acids. The building blocks of these four classes of large molecule are individually sugars, fatty acids, amino acids, and nucleotides. The individual subunits are referred to as monomers. Mono means one. And the final macromolecules, or large molecules, are referred to as polymers. Poly means many. So many monomers are bonded together to form polymers. One thing that these different classes of large molecules have in common is that they are composed of a large percentage of carbon, hydrogen, and oxygen atoms. And in the case of proteins, you also find nitrogen atoms and sulfur atoms, and with nucleic acids, you find phosphorus. Let us now turn our attention to looking within each class. So we'll start with sugars. Unfortunately, in different textbooks, sugars can be referred to by different aliases. So the first being sugars, the second being saccharide, carbohydrates. All natural sugars are made of three types of element: carbon, hydrogen, and oxygen. The ratio to every one carbon, there are two hydrogens and one oxygen. This slide shows the structural formula for the common monosaccharide, glucose. Monosaccharide means just one ringed sugar. All four of these are of the same exact molecule, represented in different ways. Glucose is made up of 6 carbon atoms, 1, 2, 3, 4, 5, and 6, plus 6 oxygens, 1, 2, 3, 4, 5, 6, and 12 hydrogens. So the chemical formula for glucose is C6H12O6. The primary chemical role of carbohydrates inside living cells falls into two categories. The first is an immediate source of energy to power the biological functioning of a cell, and the second is as a source of structural material for making complex molecules. Within cells, individual monosaccharides, as indicated here, can be combined through a chemical reaction known as condensation to form more complicated and larger structures. In this case, we have a disaccharide. Condensation itself is a form of removing atoms between two monomers, resulting in the formation of a water molecule. And that water molecule is then expelled, and the monomers are then bonded to each other by a covalent bond. In the case of carbohydrates, the covalent bonds are known by a special name, glycosidic bonds. Condensation reactions are performed by enzymes that are very specific to the monosaccharides that they join. Incidentally, the reverse chemical reaction, the breaking of bonds between complex molecules, in this case the disaccharides becoming monosaccharides, also has a name. These type of reactions that involve the addition of water are known as hydrolysis reactions. Hydro referring to the water and lysis referring to the breakage of the glycosidic bond. Turning our attention next to fatty acids, they are generally found in large quantities within the membranes of cells. However, they also have other functions, as we'll see in later chapters. The one feature molecules belonging to this class all share is that they do not mix with water. They are said to be hydrophobic, water-fearing. The reason for this becomes evident once we apply what we learned earlier in this chapter to molecules such as the one displayed here. The carbon-carbon bonds indicated between the two black balls and the hydrogen-carbon bonds indicated by the white and black ball bonds are all non-polar covalent bonds. They pull on electrons with the same intensity. Therefore, for the vast majority of this molecule, the distribution of electrons is even and there is no disproportionate distribution of charge. The very tip of this long molecule has oxygen atoms, which, as you know, are highly electronegative. They have an extraordinary pull on the electrons shared with the topmost carbon, which makes this end of the molecule ever so slightly polarized, thus the label hydrophilic. This is an example of a fatty acid. A fatty acid is a long molecule with a large hydrophobic tail and a tiny hydrophilic head. In a chemical sense, a molecule which has both properties is said to be a chimeric. And in chemistry, there's a label, and that label is called amphiphatic. Amphiphatic. We'll come back to this label in the future. It's very important that students understand. Another important distinction needs to be learnt right here with regard to lipids. The terms lipids fatty acids and fats are not synonymous. They're not the same. This molecule here is a fatty acid. Fatty acids have this typical orientation. Fatty acids combine with other molecules to form what we label as fats. And those fats are typically found in cells and cell membranes, as we'll see later in this course. The word lipids is applied to the class of molecules which do not dissolve in water. The fatty acid molecule on the previous slide is depicted here as a green bar. So in this diagram, we have 3 fatty acids attached to a small molecule called glycerol, which contains 3 carbon atoms. The resultant molecule is called a fat. Its proper name is triglyceride. 3 fatty acids attached to a glycerol makes it triglyceride. It's also known as a triacylglyceride in some books. These are the molecules that we find traveling in our bloodstream. These are the precursors to making plasma membranes, the membranes of all living cells. The reason why this slide has 2 panels is just to reinforce the principle that the shape of the fatty acid molecule may be linear, as indicated in panel A, or as panel B shows, may have a number of bends, known as kinks, whenever there are double bonds between neighboring carbon atoms along the length of the fatty acid molecule. Those triglycerides constructed of straight fatty acid chains are said to be saturated, whereas those constructed of kinked fatty acids are said to be unsaturated. This has an impact on the fluidity of structures made from these particular molecules, as we will see in chapter 11 shortly. A slight modified version of the triglyceride shown in the previous slide is indicated here. Instead of having 3 fatty acid tails, we only have 2 and sometimes just 1. The carbon atom in the glycerol molecule, which was attached to the 3rd fatty acid, has been reassigned and attached to a phosphate group instead. So the glycerol molecule once again has 3 carbon atoms, 2 of which are used to bond with fatty acids. The 3rd one is reorientated and attached to a phosphate group. Now, from your basic chemistry, you should understand that phosphate groups contain a phosphorus and 3 to 4 oxygen atoms, and those oxygen atoms are electronegative, therefore making this end of the molecule highly polarized. Further, additional modified groups can be added to the phosphate, further complicating the chemistry at this end. Once again, a single molecule which has a hydrophobic component as well as a hydrophilic component is constructed. And this is once again an amphipathic molecule. When these molecules combine together in huge numbers in the presence of water, they resemble this structure here, called a phospholipid bilayer. The reason it's called a phospholipid is simple, because of the presence of the phosphate group within the lipid molecule. All living membranes are constructed of these phospholipid bilayers. Next, we investigate amino acids, which are the building blocks of proteins and polypeptides. All amino acids have a very simple chemical structure. There's an amino group on one side, let's say the left side, and there's a carboxyl group on the right side. And there's always a central carbon atom called the alpha carbon, to which the amino and the carboxyl group are directly attached. The atom north of the carbon is always an hydrogen. However, the atoms attached south of the alpha carbon vary. And as far as we are concerned in this class, there are 20 different varieties of alternative groups which could be attached to the alpha carbon on its south side. And these we call the R groups. Amino acids are the basic building blocks of all proteins discovered in living systems. These amino acids are stitched together inside a cell according to instructions carried ultimately within the DNA. The only organelles within cells able to make polymers of amino acids are called ribosomes, which we learned briefly about previously. In this example here, we have 4 amino acids connected together by covalent bonds. Each of these covalent bonds holding a nitrogen to a neighboring carbon is called a peptide bond. Just like in sugars, we had glycosidic bonds. In proteins, these specialized covalent bonds are called peptide bonds. So this molecule is constructed of 3 peptide bonds, one here, and one down here. The ribosome selects the correct amino acid to attach to its neighbor based on the instructions carried by messenger RNA. Chapter 5 goes into much more detail about the process of translation. The final large class of molecules found inside cells are nucleic acids, DNA and RNA, and they are constructed of subunits called nucleotides. Nucleotides are interesting in that they have many roles to play within cells. One of the most important nucleotides inside a cell is ATP, adenosine triphosphate. You may recall from basic biology that ATP is a very important energy-carrying molecule fundamental to living organisms. Indicated here is a cartoon representing the construction of an ATP molecule. At the heart of the molecule, we have a sugar called ribose. Attached to the ribose on one end is a base. Attached to the ribose on the other end is a triphosphate. This molecule is able to carry huge amounts of energy in the bonds between the oxygens and the phosphorus atoms. These are also the building blocks of DNA and RNA, as we'll see in subsequent chapters. Another common theme reoccurring throughout this course and the textbook is the making and breaking of bonds reusing chemistry. ATP is a great example of one such molecule. ATP molecules are recycled by cells depending on their energy needs. In one form, ATP carries a tremendous amount of energy. In an alternative form, known as ADP, some of that energy has been released and the molecule carries less energy. However, there are systems which recharge the ADP molecule to ATP using energy from food or sunlight, and when that energy is needed by the cell to perform some work, that energy can be released. So a very important observation is that within cells, energy is stored in bonds during their formation. When those same bonds are broken, the energy is released, enabling the cell to perform some type of work. Slightly modified ATP molecules are known as nucleotides. The nucleotides are the building blocks of both RNA and DNA. Once more, the individual monomers, the nucleotides, are bonded together to form a longer molecule called the polymer, utilizing condensation reactions in the process. A special type of bond, a phosphodiester bond, is formed between the subunits. A common thread running through the formation of these large molecules is that the subunits are bonded together by specific enzymes to form larger molecules. In the case of sugars, the individual sugar subunits are bonded together to form polysaccharides. Individual amino acids are bonded together to form proteins, and individual nucleotides are bonded together to form nucleic acids. Cells also have the necessary machinery to reverse the process, whereby polysaccharides can be liberated to form the individual subunits, proteins can be decayed to form individual amino acids, and nucleic acids can be dismantled to form nucleotides. A key observation is that macromolecules do contain the individual subunits in a specific sequence, permitting cells to order an almost unlimited supply of different molecules. If cells could only put together a defined set of subunits in a defined set of ways, then there would be limitations on the types of molecules that could be generated. A general conclusion of this chapter, therefore, entails the properties of covalent bonds and the other non-covalent bonds that we talked about. Covalent bonds are utilized whenever there is a need to have something more permanent, such as individual molecules, subunits, and polymers. On the other hand, if something is needed in a transient fashion, temporarily, then non-covalent bonds are utilized. Bonding is not restricted to within the molecule. Bonding can also take place between neighboring molecules. Appropriately, bonds within molecules are referred to as intramolecular forces, and bonds between neighboring molecules are referred to as intermolecular forces or interactions. One may be under the impression that covalent bonds are more important than non-covalent bonds, but you couldn't be any further from the truth. All types of bonds are important in biology. One important fact about chemistry in this universe is that the motion of molecules is random. We heard of Brownian motion. Well, the same thing applies within cells. So when two molecules need to find each other, they do so through random motion. There's no directed force that brings them together. Simply by bumping around, they may approach each other at the right speed and in the right direction. If they do not, then they would simply bounce off and have to try again. Molecular interactions will only take place if the molecules concerned can be held together for a specific period of time. This is indicated very nicely by this figure. If molecule A needs to interact with molecule D, it may bump into molecule B. Because the surface shapes are not compatible and the intermolecular forces indicated in red are not strong enough, the two molecules will simply bounce away. The same thing would happen when molecule A accidentally bumps into molecule C. The shapes, once again, are not sufficiently compatible, and the intermolecular forces are not strong enough to keep the two molecules combined. However, should A approach D at the right speed, in the right orientation, then the shapes of D and A are compatible, as well as a large number of intramolecular forces, keeping A and D held together long enough for there to be some type of chemical interaction. Thus, once again, the shape of molecules is the most important parameter when it comes to chemical reactions taking place. A related parameter is the charge distribution on the surface of such molecules. Living systems exhibit very highly ordered chemistry. They take simple chemistry, combine that to build complex chemistry. As we've just learned, complex chemistry is obtained through random collisions. And the important thing is the shape of those molecules and the charge on their surface. Combining this observation with the fact that elements used in biology generally have small atomic radii, the covalent bonds formed within these macromolecules then tend to be strong, permitting these molecules to have a longevity suitable for living organisms. I often tell students that if I had a choice to rename this course, the course would be called Shape and Charge. Lastly, another general principle relevant to every chapter in this book is that once biological molecules are manufactured inside or outside cells, they are released into the environment. The conformation that they adopt is based on the environmental conditions, but more importantly, upon the subunits which make that molecule. They could adopt many different conformations. However, at that particular temperature and at that particular salt concentration and at that particular pH, the molecules will adopt one conformation over all other possibilities. This guarantees that in similar cells throughout an organism, say your liver, all the cells that make the same protein will have a final conformation of that protein which is identical, as long as all the cells operate under the same parameters. This module has now concluded. We should have the knowledge to address all 5 of these learning outcomes in a very in-depth fashion. Thank you so much. That concludes Chapter 2.